Well, thank you everyone for joining us today. Good afternoon and happy Friday. This is another excellent AI for Science seminar, hosted by Cornell AI for Science Institute. So many times, many students and postdocs keep asking us, how to balance the career between academic industry. And we are so grateful today that we have a distinguished speaker, Dr. Cho and Professor Cho from NYU and also from Genentech to introduce a very exciting topic on AI for molecular design. So now I'll pass it on to Laura or Yuan-Chi for a detailed introduction of Professor Cho's distinguished biography.
Thanks so much. So before I hop into the introduction, I just wanted to encourage you to participate through the Q&A if you have a question. If people have questions in the main room here, you can raise your hand and I can unmute the microphone to let you ask your question. So now just to give a little bit more of a detailed bio before we hop into the talk. Dr. Kyunghyun Cho is a professor of computer science and data science at New York University and a senior director of frontier research at the Prescient Design team within Genentech Research and Early Development. He's also a CFAR Fellow of Learning in Machines and Brain, an associate member of the National Academy of Engineering of Korea, and received the 2021 Samsung Hoam Prize in Engineering. He was previously a research scientist at Facebook AI Research, a postdoctoral research fellow at the University of Montreal under the supervision of Professor Yoshua Bengio, and received both a master's and PhD degrees from Aalto University under the supervision of Professors Juha Karhunen, Tapani Raiko, and Alexander Ilin. Without further ado, I'll pass it over to Kyunghyun to kick off the talk.
Well, thank you very much for the very generous introduction. I'm going to try to share my slide deck. And it turned out to be the perfect timing in a sense that I'm giving a talk at iClear next week. And then I thought I'm going to prepare a new slide deck that actually tells, that where I can share what we have been doing at the Prescient Design, which is part of the Genentech, which is also part of Roche. And then I think before iClear, this is going to be the first place I'm going to share all these exciting things that we have seen over the past, the 2.5 years. So today I'm going to really tell you about this, what we often refer to nowadays internally as well as externally as the lab-in-the-loop therapeutic antibody design. And then this is going to be me telling you about the progress that has been made, not only by me, but by the entire team of Prescient Design, as well as the entire group of Roche. So because this is going to be a bit of a rehearsal or the test run of the iClear keynote as well. So I have some pictures. By the way, I don't know if how many of you are familiar with the conference iClear, International Conference on Learning Representation, which has now become one of the major conference or the publication venues in machine learning. Only started slightly over a decade ago, and then it started in 2013. The first version happened in Arizona, Scottsdale, and I was there. And then back then, there were actually more cactus than conference participants. Now, of course, it's really difficult to imagine how much it has changed, but I just wanted to tell you this quickly. But anyway, so AI for protein design, that's what I'm going to talk about today, in particular, focus on antibody. But obviously, AI for protein design can be done relatively easily if we believe in all those, the excitements about the large-scale language models. Right? But let's go back to what we mean by AI for protein design, and then what it would look like in this era of large-scale language models. So what will we do? We're going to put a transformer in the center, and then we're going to train this transformer language model on a massive amount of data that contains both proteins as well as the natural language descriptions of them. And once the training is done, we're going to simply ask this language model, just like what we are doing with the ChatGPT these days, to design some proteins. So for instance, I might ask this language model to design a metal-binding protein for me. And, I've been told over and over by a lot of my colleagues as well as the friends that I have to be very polite. And this language model is going to give us the metal-binding protein. And as Ilya Sutskever said in 2014 at NeurIP's talk, success is absolutely guaranteed here. So in fact, I could very well just finish my talk here and then talk about the AI for protein design in the context of the language models. But that'll be too easy, right? So instead, I'm going to try to go into the small details and there are a lot of, let's say, issues as well as the problems that we still need to solve going forward. So first thing I need to remind you of is what protein is, and then often you all have taken, I mean, I do notice that there must be some biologists here as well as many of the other scientists. So I'm going to give the simplest possible picture here is that a protein protein starts from the DNA, which is transcribed into RNA, and the RNAs are going to be translated, or the messenger RNAs are going to be translated into proteins. And then this sequence of amino acids will fold almost spontaneously into a 3D structure that actually performs some kind of function. But here the point I want to make is that a sequence of amino acids, in fact, encodes the structure. And this sequence consists of varying number of the amino acids. Now, there can be a lot of different amino acids, but often there are 20 to 22 amino acids that are encoded by genome. And then this sequence determines the structure as well as dynamics in combination with the environment. Now, once the protein folds into a 3D structure, these proteins interact with each other as well as the other molecules in order to perform certain biological functions. And then what this actually tells us is that we can now design a protein to interact with particular target molecules in order to induce some desirable functions artificially. Then, of course, as a computer scientist myself, my mind goes immediately into the simplest or the most simplified setup. That is that the protein design must be just nothing but a sequence optimization problem. So what the goal is that given some objective function f, we are going to find a sequence of amino acids S that's going to maximize this objective function. Sounds simple enough. And indeed, this is the key idea behind the so-called directed evolution, which received the Nobel Prize in Chemistry in 1998. We start from some known sequence. We're going to apply the random mutation, producing many different types of the mutated sequences. And then these sequences are all going to be tested in a high-throughput screening. And then we choose only the top best sequences. And then we repeat this process over and over until we find the sequence that's going to have or exhibit a very high value of this objective function. And then, in fact, in addition to this kind of idea of the directed evolution, there are so many wonders of biology as well as of biotechnology on top of which protein design sits. Because I'm not a biologist, I'm not going to go into any of those details, nor am I qualified to talk about it too much, but I just wanted to list up few ideas that are just amazing from my perspective, and the few ideas that actually we use directly every day in order to perform protein design. These include PCR, recombinant DNA, GFP, as well as the CRISPR-Cas9. And unsurprisingly, all of these have been the reason why the Nobel Prizes were given out during the past, let's say, several decades. Now, biotechnology, it works amazingly well. And now, if we want to do the protein design using artificial intelligence on top of these wonders of biotechnologies, what are the challenges? There are 3 challenges that I want to talk about today. I'm going to go into detail each and every one of them. But then the point I want to make here is that the goal is not going to be to give you the full set of the solutions that are going to tackle these challenges, but to tell you these challenges exist and that our research really has to be driven by these real-world challenges that are there in order for us to solve real-world problems. And before I continue, I gotta give you a couple of warnings. The first thing is that I'm going to talk about a series of obvious things. And in fact, some of you might think that for some parts, what I'm saying is so obvious that you might find it somewhat boring as well. But I'm going to do so because it turned out that it's all about putting all those obvious things together in order to solve a real-world problem. And then the second warning is that I'm a computer scientist by training. Only courses that I have taken that are relevant, even remotely relevant to biology, are the advanced physics, basic chemistry, and general biology during my undergrad years. And then let me tell you, I did not do well. So if there's any biology-related questions, either I'll have to refer you to the biology textbook or some of my colleagues who may be on the call as well. So let's talk about the first challenge. That is a computation versus biology. In designing proteins, there are two processes that must work together. First one is computation, and then the other one is experimentation. Computation algorithms are going to be used by us to produce new designs of proteins. And on the experimentation side, these newly produced designs are going to be synthesized and tested. And then the results of the experiments will be fed back into the computation. Unfortunately, these two processes cannot really work well in synchrony. What do we mean by that? On the side of the computation, we can always improve the throughput as well as lowering the latency. How do we do that? We call Jensen 1. If we call Jensen 1 over, we get more and more GPUs, and then we can run our algorithms faster as well as at a higher rate of the distribution or the parallelism. And then this allows us to just churn out infinite number of potential protein designs. On the other hand, experimentation cannot simply be sped up. In fact, in bacteria, in a very idealized situation, it takes about 10 seconds to transcribe a 1,000 base pair long gene into RNA. And this is extremely slow, if you can imagine this. With the 10 seconds, we can run, let's say, our protein design models on hundreds of GPUs to produce millions of the candidate designs. But there's no way we can actually test them with the experimentation because of this gap in the rate. So how do we actually overcome this difference? Of course, this is not a new idea. This is not a new problem. Already from 1990s, we have known about the idea of the sequential model-based optimization. The idea is that instead of running the experiments over and over, we're going to have a machine learning surrogate function that approximates the outcome from the experimentation to a certain degree in a local region. And based on this machine learning classifier, we're going to optimize the sequences in silico before we send those selected sequences to a lab, very rarely. And this can be thought of as a two-stage process. In the first stage, that is called in silico optimization, we use this machine learning surrogate function f in place of the actual experimentation. After running it for many iterations, we're going to choose the top K prime candidate sequences, very small number of them, and then send them to the lab. Now, only this small number of designs are going to be tested, and then the feedback is going to go back into the in silico optimization in the form of the training data for the surrogate function, as well as the starting point of this in silico optimization. And then we can iterate repeat this process over and over. And indeed, in many of these reference problems, the real-world problems as well as the synthetic problems, if we have a good surrogate function, such a model-based optimization can give us an amazing boost over repeatedly querying the lab directly. And then when I say boost, I'm talking about boost in the acquisition values, the objective values, with respect to the number of the actual queries, where each query corresponds, you can imagine it as running the experimentation at the lab. This sounds so good, and then this sounds like already every problem was solved in 1998, but in reality, that's not really true because the objective functions that we need to work with tend to be non-decomposable. That is that we cannot really write down the objective function as the sum of the individual sequence objective. Why is that? We can imagine a lot of different scenarios, but few examples where these objective functions are not decomposable include diversity. We often want to test as diverse set of sequences as possible, but then of course computing a diversity requires us to look at the entire set of sequences. Another example is a multi-objective optimization that I'm going to get to shortly. Furthermore, we cannot even send an arbitrary number of sequences to the lab. Even if the lab is moving really, really fast, it doesn't mean that it can process as many sequences as we want to send them to. Instead, there is a severe constraint on the cardinality of the set of sequences that we can send to the lab each time. Now, this constraint comes from many reasons, including budget. The labs are not going to run automatically for free. Of course, we need to have people. There are issues of the supplies. And then, of course, there is a strong and then hard constraint that is set by biology itself. So then what do we do? The simplest thing that you can imagine is a greedy selection. We're going to consider all possible sequences in the world. And then we're going to consider one sequence at a time, in addition to all the sequences that have been selected already. So we're trying to find the one sequence that's going to maximize the improvement in the overall objective had it been included into this selected subset. And then we continue doing so until the selected set B becomes as large as the target or the constraint set by the lab. Unfortunately, this is intractable because there are just so many sequences that we need to consider each time. So instead, what is much more realistic is that if we do the greedy selection, however, within a finite proposal set that was given to us. So instead of looking at all possible sequences, we look at only the sequences that were given to us by some other mechanism. Now, obviously, the big question and then the big criterion that needs to be satisfied here is that the candidate sets must contain a lot of good designs. And what we do, we actually can use a generative model to produce a good candidate set. And I'm going to talk about that near the end of my talk later. Of course, we are all using deep learning these days. And when it comes to deep learning, there are many different ways to define it. But in my view, deep learning is where we use gradient descent. That's essentially my view. And then what that means is that if our acquisition function is differentiable with respect to the input sequences or some representations of them, we can now use this objective function to guide the generation of the sequences directly. And then, in fact, this idea, that is to replace discrete search with optimization in a latent space, was the key idea behind the founding of Prescient Design in early 2021. And I'm going to again talk about this a bit later. Now, unfortunate thing is that, of course, we often cannot choose the objective functions ourselves. Objective functions are given to us by the necessity of the design. So why are we designing proteins? And then the why part is not the one that we just decide. Why is actually decided by the external factors. We want to design proteins for the purpose of therapeutics. We want to design proteins in order to improve the various synthetic biology pipelines. And there are so many different reasons, but we don't get to choose them. The real world chooses those objectives for us. So then, obviously, we cannot really force the objective functions to be all differentiable. In that case, one obvious way to go about is to train a policy or a stochastic sampler so that whatever the sequence we get out of maximizes the objective function on its own. And once we have that policy, we can just sample k prime many sequences and then say that that is going to be the set. Now, as you can imagine, this is not the greatest way to go about because it doesn't really take into account the relationship among all those sequences within this subset B. So instead, what we can do is we can now train a greedy policy directly, and then we're going to call it a set condition policy that looks at what has been generated and then tries to generate the next sequence that's going to maximize the improvement in the objective function. Again, this is not the optimal way to go about because the optimal way is to actually go through all possible combinations. However, tractability in the real world is super important. So we're going to stick to this greedy policy and then we use the reinforcement learning again to replace discrete search. And it turned out that indeed doing so really helps optimization. So starting from the model-free optimization, moving to model-based optimization, and then to the independent sampling under the cardinality constraint, and all the way to the trainable set condition policy, we do see that we can build a better and better black box optimization algorithm. And this really tells us a lot. At the end of the day, what is this black box optimization algorithm that we are using for? This is the interface between fast computation and slow biology. And then because this gap is so high, we have to make sure that our black box optimization algorithm takes into account all possible aspects that are involved in this gap, and then so as to make the optimal choice. So I just showed you that this is a problem, and then there is a way to go forward, but this is not it. We need to go even further. We have to think about how to make a better trade-off between sample complexity and accuracy from the computational perspective. But also at the same time, we have to work on how to improve the throughput of the experimentation itself as well. So this was the first challenge. That is that when it comes to protein design, that is, or the real world material design, there's always going to be a big gap between the computation and the real world. And then we have to make sure that we know how to narrow down this gap as much as we can, either algorithmically or by some other means. And it turned out, unfortunate thing is that even if we can do the black box optimization well enough, that's not enough because we never get a single objective function. Let's imagine designing antibodies, and in particular the therapeutic antibodies that are supposed to help, let's say, people from having various adverse conditions. First thing that we can imagine is that, of course, we need to be able to, we want our antibodies to be easily synthesizable. If we cannot synthesize them at scale, it really doesn't matter how amazing the therapeutic antibody that is. If we cannot produce them, manufacture them at scale, who actually cares? And also, another way to put it is that the antibodies that cannot be expressed are not antibodies. It's just a hypothetical dream that we have. And obviously, expression alone is not enough. These antibodies need to bind or interact with the target antigen, and only the target antigen in order for them to perform biological functions that we want these antibodies to perform. Now, unfortunately, this is not all either, because anytime we inject anything to our body, our immune system is going to respond quite rapidly. And then sometimes this response can be quite severe. So what we want is that we want to design an antibody that expresses well and that binds really well to target antigen, but does not invoke severe immune response. That said, are these all? No, we actually need to think more stuff. For instance, if we cannot deliver these antibody therapeutics to the right place, it doesn't matter whether we design the amazing antibody therapeutics or not. So we must be able to deliver it to the right parts of the body. And after that, we want the antibody therapeutics to clear off the body as well at the right rate. Otherwise, it's going to have a lot of the side effects that we do not want these patients to suffer from. So there are so many properties, and some of these properties may very well compete with each other. And then this really brings up a difficult problem. And then these are multi-objective and not sequential optimization problem, because these objective functions often have to be taken into account simultaneously from the very beginning. I just made a very simple animation of why it's really important to consider all these objectives all together from the very beginning. Let's say what you're seeing on the screen on the right-hand side is the space of all the antibodies. And then you're seeing some combination of the two objectives or the existing objectives that we know how to compute very easily from the very beginning. And then it turned out that there are two local minima. And then the one on the top right side is a much better local minimum. So if you run some kind of optimization algorithm, we're going to find all those amazing antibodies that are concentrated on the upper right corner of this space. However, it may be that we find that we need to optimize for yet another objective function that looks like this on the right-hand side, that looks almost like constrained. So it's 0 almost everywhere, half of the space, but then suddenly it jumps to 1. If we are suddenly trying to maximize this particular objective, starting from the earlier ones, these red dots, the best we can do is to go to the place that is high enough according to this new objective while not moving too far away from it. But that point, the region where these red dots have actually arrived at are completely nowhere. We shouldn't design any antibodies like that. In fact, if we started from all of these objectives from the beginning, we would find a good enough, let's say, solutions eventually that are also feasible. So we really need to think of all these objectives simultaneously from the very get-go. And second reason why it's really difficult is that we actually don't know what is the right balance among these properties should be early on. So you can often the example that I take is the balance between the safety and then efficacy. So sometimes some of the drugs are not as safe as other drugs, but may have a much greater efficacy and vice versa. Then what is the right kind of trade-off? We actually don't know. That trade-off is going to be decided down the road when we run in vivo testing as well as clinical trials and whatnot. What that means is that early on, when you design these molecules, we have to explore the entire spectrum of the different trade-offs. That is, we need to explore the parallel frontier. And this all actually boils down to the problem of measuring the quality of the frontier. And then there are many possible ways to measure the quality of the frontier, and in particular in this kind of multi-objective optimization community, people have come up with many different ways to measure the quality of the frontier. Some are based on volumes, some are based on the random scalarization, some are based on more information theoretic metrics such as the entropy. And then once we choose one of these quality metrics, it becomes effectively equivalent to single objective optimization. And at Prescient Design, we've been using expected hypervolume improvement, or its variant, noisy expected hypervolume improvement, for some time. And hypervolume is an extremely intuitive concept. As you can see on the right-hand side of this slide deck, if I have all those points in the objective space, what I want to know is that what is the hypervolume that is taken by these points that I have selected with respect to the reference point. And then what I want is to maximize this volume. And then this volume means that I cannot simply maximize one of the objectives, but I always need to look at all the objectives all together. And then this objective has been actually quite successful for our purpose of the protein design. In fact, our scientists, Sam Stanton and others, have shown last year at NeurIFS that by maximizing expected hypervolume improvements, we can really improve the parallel frontier with the extremely high coverage of all possible combinations. And then this really applies to real-world antibody design as well. When we maximize the expected hypervolume improvement for antibody design, starting from the 20 seed antibodies for 4 target antigens, after 3 rounds, we found that the 43% of the designs expressed a higher yield than the seed antibodies, and then 21% of them exhibited a better affinity. And then these are all the experimentally validated and synthesized examples that I'm telling you about. So expected hypervolume improvement seems to be a good acquisition function or a metric to measure the quality of the parallel frontier. However, we can always do better because there are always some issues to be resolved. And one of the issues with many of these multi-objective acquisition functions is that they are scale sensitive. And when it comes to objective functions you run into in real life, we never know what are the right scales to use. What is the right thing? Can I just put the log there? Can I exponentiate it? Can I square it? No one really knows. Scale is a big issue. Can I just multiply with some numbers? No one really knows. And then what this is equivalent to saying that we don't know the right balance. While we actually use a particular scale for one objective, while some other scale for the other objective. And as you can see on the slide, on the screen now, if you use a hypervolume, the choice of the parallel frontier would change if we apply some different transformation to one of the objective functions. And then this really poses a dilemma. I mean, which one is better? Is the left one better or the right one better? No one really knows. So instead, another direction that we've been pursuing is to use a ranking-based acquisition function. So instead of looking at the actual values, we're going to fit the cumulative density function on all the measurements we have, and then trying to find the level set or the level set with the highest value of the CDF, and then use all the points that lie on the level set as the Pareto frontier or the approximation to the Pareto frontier at the current measurement level. Instead of talking about the details, I think this figure shows you the whole story. So on the left side, we have all those measurements. There are seven of them. And then we're going to feed a cumulative density function. And then what we see is that those that are ranked equally are going to be on the same level set. And then the goal here is to find the ones that are in the outermost level set. And indeed, using this bow tie, that's what we call it, the Bayesian optimization with tight multivariate rankings, we see that there is no issue or the issue of the choosing any particular scale for the objective functions. It's all about just ranking. Now, at this point, if there is any statistician in the room or somebody who's been working with the cumulative density function in the room, they'll just raise their hand and then ask me that, but how do you actually estimate the CDF? Because that sounds like actually even more difficult problem than solving the problem of finding the Pareto frontier. It turned out that we can actually rely on the pretty old concept of the copula and the Vine copula in order to very efficiently estimate or approximate the CDF. And then indeed, what I'm showing you on the right-hand side of this slide deck or the screen is how you can actually do so. So fit a Vine copula and then evaluate CDF using literally 5 to 6 lines of Python code. And it turned out, indeed, doing so improves the Pareto frontier very rapidly, even when it was measured by the hypervolume, which is the one that is used by the hypervolume improvements as improvement acquisition function. And furthermore, even more important is that the computational overhead does not increase as rapidly, even when the number of properties grow. As I told you, our goal is to take into account as many properties as possible from the very beginning. What that means that the scalability with respect to the number of parameters is really important. And then this kind of bow tie or the bow tie allows us to scale up the number of the objectives very readily. Now, not everything is rosy here because this bow tie is not differentiable, unlike the expected hypervolume indicator or the improvements, and it cannot be easily integrated into a neural net generator. This is a bit of an issue that needs to be resolved over time. So I showed you that yes, multiple objectives must be taken into account altogether. And doing so, we have to come up with a good acquisition function. And then once we have a good acquisition function, we can rely on the black box optimization from the first part to find the set of designs or the sequences on the Pareto frontier. However, there are a lot more issues that needs to be resolved. It's just so difficult to solve a real-world problem. There are so many details. One of such challenges is a known dependencies among the properties. A lot of the properties that I told you about and then I'm going to run into over the past next few years are not independent quantities or the independent objectives. They actually interact with each other. For instance, if the sequence cannot be expressed, sequence cannot bind to anything else because they don't exist. If the sequences don't express, they cannot be cleared nor delivered because they don't exist. So what is the right way to capture these dependencies and impose these dependencies on designing an acquisition function or trying to compute the acquisition function more efficiently is a big remaining challenge. And then the second challenge, which I think is more interesting and more difficult, is the fact that some of these properties are simply too costly to measure. And then what that means is that the availability of these properties dramatically vary across different properties. And also, some of them will require a significantly more investment in order for us to measure them. What that means is that there is a vast amount of difference across these properties in terms of the utility. So can we actually take into account the investment we need to put in order to collect the measurements into the acquisition function or the black box optimization in order to really make a better trade-off between sample complexity and accuracy? And then there are so many issues remain. So somehow I feel like, up until now, everything is just difficult. However, that's the thing, is that the real world problems require real world challenges, and the real world challenges requires actual innovations. But at the end of the day, it's all about putting them together. So I'm getting there. So stay, be patient with me. So the third challenge is the fact that the desirable proteins are few and far apart. The space of proteins is gigantic. If you think about all possible biologically relevant proteins of length, let's say L, there are like 20 to the power of L many different sequences. And what that means is that the problem of protein design is equivalent to finding a needle in a haystack, and then this haystack is a gigantic haystack. So we must narrow down the search space significantly. And in doing so, we have two sources that we can rely on. One is biology itself, and the other is the property predictor. I'm going to go through each one of these more carefully now. What is really amazing is the fact that there was an algorithm. There is this algorithm called evolution that has resulted in us, and that is constantly running in the background. And the evolution has been running over and over, nonstop, resulting in a millions or billions or trillions of the different proteins that have been tested. And then we have sequenced millions and billions of proteins from nature, creating these gigantic databases of protein sequences. For instance, BFT has more than 2 billion protein sequences. UniProt has more than 200 million protein sequences. And these are footprints of evolution exploring the space of proteins. And what that means is that perhaps we can rely on what evolution has done in order to narrow down the search space to the vicinity of the evolution's footprints. And that's exactly how we started whole Prescient Design. So Prescient Design started as the spin-off from NYU. And then already in 2019 and 2020, the two other co-founders and I, so Rich Bono, Vlad Grigorievich, and I, were working on this what we call function-guided deep manifold sampling. So we're going to use a massive amount of proteins that were explored by the evolution that are stored in the protein sequence database to train a denoising autoencoder to capture the protein manifold. And then we will explore over this protein manifold that is guided by the function classifier. And then this idea actually worked pretty well in 2019 and 2020 already. We tried to see if we can use this kind of model to reintroduce a removed metal binding site. We could do that. We wanted to see if we can actually improve the function of a protein with a known function. In this case, we're trying to maximize the chance of exhibiting the cutinase activity. And then we could do that. The amazing thing was that all these sequences were very, very different at the sequence level from the original protein. And then we wanted to see if we can easily swap or jump from one type of the protein to completely different types of proteins. And then it turned out we could do that as well. And that's the reason why we started the Prescient Design. And then eventually we became part of Genentech. Now at Genentech, we are focused on antibody design, as my talk title actually indicated. And when it comes to antibodies, it turned out that there is yet another source of information that we can rely on in order to narrow down the search space. And then that is the adaptive immune response. You can almost imagine this as a real-time evolution in our own bodies. Whenever some foreign agents are introduced into our body, our immune system is going to react to those antigens. And as you can imagine, everyone, depending on where they are and what kind of, situation they are in, they're going to be exposed to different types of the antigens. Now, our immune system is going to adapt to these different types of the exposures, resulting in a diverse set of the antibodies that are human-like and are often very, very useful. So the OAS, Observed Antibody Space Dataset, that is maintained by Oxford, for instance, contains more than 1.5 billion antibody chain sequences, and then more than 120,000 pair chain sequences. And now we can rely on this in order to narrow down the search space of the antibodies. And indeed, this is actually the paper that we're going to present at iClear next week. So Nathan Fry and other trained discrete walk-jump sampler on a small number of binding antibodies, about 10,000 of them, produced from trastuzumab, that is the Herceptin, using CRISPR-Cas9. So, and then this data set was created and released by the Mason in 2021. Now, I'm not going to talk about discrete walk-jump sampler too much. You can actually use your phone to take a picture of the QR code and then check out the paper and everything. It's much better explained there. However, the basic idea underlying the discrete walk-jump sampler is to separate, but then learn simultaneously to extract good representation of discrete sequences and sample effectively and efficiently from this representation space. So we call this learning of the representation, and then going back and forth between noising process and then denoising as jump, and then sampling in this representation space as walk. Once training is done, it's extremely easy to iteratively sample sequences or the discrete objects in this representation space by taking a walk, jumping back to the clean version, taking another walk, jumping back to the clean version, on and on and on. And because this model was trained on these antibody sequences, what it's going to generate is all those antibody sequences that are very likely or similar to the ones that bound to HER2 antigen, but are not exactly the same. So we use this discrete walk-jump sampler for redesigning the CDR, that is a complementarity determining region of antibody, and up to 10 mutations. Now, 10 mutations of the amino acid sounds pretty small. However, if you think about how many possible mutations there could be, there are essentially 20 to the power of 10. It's not a small number. Is it like the impossibly large number? Not really, but also it's not a tiny number either. But then we trained this walk-jump sampler and drew 1,000 samples alone. And out of the 1,000 samples, 70% or so of them bound to HER2, and then the binding affinity was as good as or close to trastuzumab, which is in fact the FDA-approved antibody therapeutics targeting HER2. And almost all these sequences were unique. So essentially, walk-jump sampler was not trying to produce a one good thing over and over, but it was able to explore the vicinity of the binding antibodies. Unfortunately, even then, space of proteins is still too large. What that means is that not all amino acids matter for improving some of these properties. In the case of the binding, we know that the amino acids that are in these complementarity determining regions tend to matter most. And then there is a reason why in the previous slide, I told you that we tested mutating only the CDRH3. But then for other properties, we actually don't know. And if you think about how long these antibody sequences are, it's actually hundreds of the amino acids. And then we probably don't want to, in fact, mutate all those things. We only want to mutate a small number of them that are relevant to the properties that we want to maximize or minimize. And what that says is that we cannot really do that in purely unsupervised way. So what we do is we rely on the saliency map. And whenever I think about the salience map, I feel like I'm back to like my my postdoc years in 2014, when Karen Simoyanan and others showed that they will take this gigantic convolutional network, and then it turned out that for each of the images, we can tell what are the most important pixels for making prediction. That's the one that you're seeing on the right-hand side. And that's from 2014, pretty incredible, right? That's exactly 10 years ago. However, it turned out that in 2018, Julius Adebayo, who was a postdoc with me at Prescient Design until last year and is now a CEO of his own company, Gaudi, he and his colleagues show that indeed the salience maps are visually appealing, but they are not necessarily faithful to the data generating process or the actual classification process. But then, fortunately, last year, before Julius left for his own adventure, he has actually noticed that with proper regularization, the salience maps start to become more reasonable and more faithful to the data generating process. There's definitely a more research to be done in order for us to explain this much better. However, empirically, we found that having a proper regularization, such as the independent Gaussian noise or the adversarial robust optimization, almost always led to a classifier that is able to identify the correct places that are relevant for making a prediction. And then what happened is we were in fact using it in our lab in the loop design. We trained these property predictors that are used to compute the acquisition function, but also we use them to figure out what are the amino acids in this sequence that matter most for a particular properties that we are predicting. And then what you are seeing on the left side of this slide, this screen, is that the salience map have correctly identified these CDR regions based on the classifier that was trained to predict the binding affinity. And then by focusing on mutations in these smaller regions, we can improve the acquisition value so much more effectively and rapidly than considering the entire sequence, because we can avoid any of those spurious correlations that exist that are not relevant to the actual target properties. So I just showed you that, okay, some recipes in order to build this kind of good generative models to explore the vicinity of the narrow down search space in the protein space. However, I have to emphasize just once more that the combinatorial optimization is by definition very, very difficult. And then it is just due to the curse of dimensionality. We cannot really get away from this one. And then in order to cope with this, at least, we must recruit help from everywhere we can. And in our case, we get help from the evolution, we get help from the adaptive immune response, we get help from the property predictor's ability to capture correlations, and hopefully down there was a bit of a causation as well, causations as well. And then finally, we use the lab in the loop platform to iteratively collect more and more data. But even then, it's a very difficult problem. So if you put all those things together, we end up with this lab in the loop therapeutic antibody design platform. We have the central database that contains everything, to which we refer as DesignDB. There's a generative models of the proteins that are going to produce a bunch of proteins, and these proteins are going to be scored by the property predictors, and then these property predictors, the scored sequences are going to go to the multi-objective black box optimization algorithm. And then this optimization algorithm is going to produce a small subset of the designs to be tested, synthesized and tested in the lab. And then once the experimentation is done, the feedback goes back directly into the DesignDB. And this is what we mean by the lab in the loop antibody design. Now, unfortunately, this is not enough, sadly so yet. We have to start from somewhere. So we often rely on many other more traditional ways to design antibodies, such as the immunization campaigns. At this point, you have to ask yourselves, and then you probably should ask me, whether it actually works. The answer, of course, is going to be yes, but I'll show you how well it works. But before getting there, I gotta tell you that I only told you about 2/3 of what is needed in order to build this kind of lab in the loop design system. What are those 2/3? AI/machine learning, biology, and experimentation. Okay, maybe 3/4. But then there is one thing missing that I didn't tell you, is that what is actually driving this lab in the loop design platform? And it turned out the main engine behind this lab in the loop design is software and hardware engineering. So we need to have an extremely well-done compute infrastructure. We need to have an extremely well-done infrastructure software that supports training, serving, and monitoring of machine learning models. We need to have an extremely well-done data management system. We need to have an extremely well-done lab integration. And then we also need front-end interfaces in order to support various scientists and engineers. So in fact, Prescient Design is an engineering first drug discovery team. We do have, of course, a molecular design team. We do have a frontier research, which is focused very much on the research. But then a main part is the engineering that consists of the infrastructure engineering, data engineering, and machine learning engineering. So what we have built, this is a whole system we have built. This is a system that we can really click and then work. It exists on top of this compute infrastructure. So there's a design DB, and then the sequences in the design DB are going to be used to train these generative models. And then these generative models are going to produce the new designs based on those old designs. And then these new designs are saved in the design DB, and these new designs are going to be scored by property predictors. And then these property predictor scores are also stored in the design DB. And multi-objective black box optimization pulls all those new designs with their scores from the design DB and then send a subset of that to the lab. And then once the lab finishes the experimentation, all this feedback will semi-automatically come back into the design DB. And then we run it over and over. And then why does it have to be, and then why is it loosely coupled? Because at every stage, we're going to have the actual expert in machine learning, expert in biology, expert in chemistry, expert in physics, expert in software engineering, all looking into what are being passed through different stages in order to ensure that we are not making any unnecessary and also wasteful designs. So we've been doing this for about 2 years now, since August 2021. Actually, it's like closer to 2.5 years. And then because what we are trying to build is not just therapeutic antibody, but a platform from which we can design the therapeutic antibodies over and over, we've been working with more than 15 target antigens. And then we've been working with the many different methods that have all been integrated into this lab-in-the-loop platform. I showed you the Lambo, that is the classifier-guided discrete diffusion with the hypervolume improvement. I told you about target-specific walk-jump sampler, but there are quite a lot more methods that are yet to be disclosed. And all these methods produce sequences that are going to go into the global selection by the black box optimization algorithm. And then only a small subset will be selected at each round to go to the lab. And over the 2 years or so, we have gone through more than 12 loops of this design. And then these loops were there for both development as well as design. So I'll show you the actual example. All right, so I cannot reveal the target names, unfortunately, otherwise our legal team is going to tackle me while I'm telling you about this. So I'm going to call it target A first. There are going to be 3 targets that I'm going to show you about. Lambo is the classifier-guided diffusion with the hypervolume improvements. And then for this target, Lambo was run for four loops. So I don't know why it says 3. At the very beginning, it found designs that are both not expressing enough and are not binding well enough. However, in the next round, this algorithm or the lab in the loop realized that we can improve the expression quite a bit while maintaining the affinity. And then after that, it realized in the subsequent rounds that it's possible to maintain the expression, but improve the affinity as well, resulting in a nice frontier that covers both the high expression, low binding to the high binding but low expression antibodies and in between. And obviously, Lambo is just one of the methods. We use so many different methods, as you can see here. And then what we end up with is that every time we run these rounds, we're going to push the frontier further and further up toward the top right corner. I'm showing you only 2 major properties that we care about, but of course, there are many other properties that we consider as well. Let's look at second target. Same thing, Lambo. In the first round, it found a bunch of high expressing to a low expressing antibodies, but they were not really good binders. But in the next round, it realized that it could actually improve the binder a bit if it sacrificed or at the cost of the expression. And then based on all those things, in the next few rounds, it figured out that by sacrificing expression further, it could design all those much better binders. Again, covering the entire frontier as well as we can imagine. And then if I look at all the other methods together with the Lambo as well, what we see is that in round 7, we were covering high expression modular binding region of the space. But then we started to figure out that the binding affinity can be improved at the expense of the expression yield, eventually resulting in extremely high affinity binders, but that are not as expressing as the earlier ones. So the target C, this one I want to just show you the whole picture first. And these R3, the one on the bottom right corner corresponds to seed antibodies. So we started with some seed antibodies that are highly binding. However, they do not express that well. And then we started from there. We asked all these different methods to design new things. And then we collected all of them and then put it into a black box optimizer, went through multiple rounds. And then what we see is that in the next round, in the round 9, we found a lot of extremely high expressing sequences, but with a moderate, let's say, binding affinity. And then after that, this method, our lab in the loop, figured out how to interpolate between these 2 points. Because at the end of the day, no one knows what is the right balance here. Most important thing is a good coverage of the good combination of different properties. And then you do see that this lab in the loop design process explores this prior frontier over the multiple rounds of design and experimentation. So the answer to the question that I asked you is that yes, we can design antibodies. And this part of the design will be called hit-to-lead generation and also the lead optimization. We've tested with a number of target antigens. We test with a flexible number of machine learning algorithms as well as the first principle-based filters. And then, as you have seen, our platform explores the prior frontier of the multiple objectives over multiple rounds extremely efficiently. However, it's too early to pop the champagne yet, unfortunately. Why is that? Because I just told you about the protein design. And protein design is not drug discovery. What is drug discovery? Drug discovery is a multi-year, actually multi-decade effort that starts from the science that tells us about the diseases or the conditions. That is followed by the target discovery. So what are the biological targets that we need to design the drugs for? And then once we know the target, we need to now design the molecule. And then these molecules need to be optimized and characterized before we can test them in vivo. And then only after the in vivo validation is over, we're going to slowly test it on the actual humans, that is the clinical studies. And only after that, these are going to be prescribed to the actual patients in the clinic. And as just an example, we all heard about the Wegovy and then how FDA approved the Wegovy's expansion to reducing the excess body weight. That just happened a month and a half ago. And then that's 2024 March. But then if you think about it, okay, what is Wegovy? Wegovy is just a brand name for the semaglutide. And semaglutide was originally designed to treat the type 2 diabetes. But then when did we actually figure out the molecular basis of diabetes? We actually go all the way back to 1999 to 1921. And in particular in 1921, Banting and Best at the University of Toronto discovered or isolated insulin. And then that turned out to be the insulin that we are all talking about. And then in 1980s, as part of this efforts to treat the diabetes, the GLP-1 was identified by the Hebner et al. And then Mosov. And 20 years later, or actually slightly more so, the FDA finally approved the very first GLP-1 agonist, exenatide, for type 2 diabetes. And then from there on, it took another 15 years for people to have developed the semaglutide. And then another 3 years for this use case to be expanded. And then what that means is that this whole drug discovery is not a one-shot or something that we can say that, well, every 3 years we're going to have a new drug or anything like that, or every 4 years, my vesting period is over, let's go move to another company and then make another drug. It's a long-term process. And then what I showed you today covers about 1.5 steps in this long process. So when it comes to drug discovery, there are so many things that we need to do. And then what I showed you is a promising step, but it's a promising tiny step that we have taken. So then, in my view, this is going to be completely my view, what is the true goal of AI for drug discovery? Not for molecular design, but for drug discovery, is to be able to make a prediction all the way to the clinical outcome from every stage of this drug discovery, and then be able to backpropagate predicted clinical outcome all the way to the coordinates of the atoms or the target discovery stage and whatnot, in order to maximize the success rate, so that even if it took 50 years to make a drug, if you know that the success rate is higher than 10%, which in my opinion at the moment is less than 0.1%, that's going to change what we mean by doing drug discovery. And then that actually almost completed my talk, but one thing I can tell you, I told you that the Prescient Design is this and that, but in fact, it's not just Prescient Design, but it's a pan-Roche initiative. There were teams from antibody engineering, next generation sequencing, computational catalyst, structural biology, biochemistry, the properties, PRED, diagnostics, and even legal team were all work together with the Prescient Design. And then Prescient Design is now about 70 strong, let's say, organization that consists of the engineers, machine learning scientists, as well as biologists. And this is actually the end of my talk. Thank you.
Not sure how well you were able to hear the applause, but hopefully you could hear that. So wonderful talk. Thank you so much. Are there any questions from the room? Great. I'm going to walk over with the microphone.
Thank you so much for the excellent talk. I'm actually very curious about it. Maybe it's over detailed. Like when you say Pareto optimality, when you want to optimize it, is it differentiable or how slow? I mean, I can imagine because you need to generate a set and then you calculate it, and you need a surrogate model and you need to back propagate through all those. Yeah. So is it something that's efficient to do? Oh, sorry. I think you're muted.
Sorry about that. Yes, computing the quality of the frontier and then trying to see whether maximizing that's going to be a Pareto frontier is a question that we need to ask for every acquisition function that we come up with. But the next question is actually exactly what you ask, is that is it really doable to maximize that acquisition function? So I can actually talk about one of them, bow tie, that I just talked about earlier today. In the case of the bow tie, it was true. So it's very clear that what we want is this kind of ranking-based criterion from our perspective, because many of our objectives have a very arbitrary choice of the scales. So we wanted to go with the ranking. And then, of course, once we started talking about the ranking, we could talk immediately imagine, oh, we can use a cumulative density function. But then the question is, can we actually compute the cumulative density function in a tractable manner? Not really, because it requires us to solve a lot of integrals. But then it turned out that Natasha, who is the second author of this paper, has been working with this kind of the idea of the copula for the uncertainty quantification earlier on when she was doing her PhD. So she was able to connect this concept to more efficiently compute the CDF. And then that actually allowed us to compute the bow tie relatively straightforwardly. Now, unfortunately, it's not really differentiable because it's a ranking criterion on this levels. So across all those level sets, it's actually a discrete jump. So there is no gradient there. So we are actually thinking about how to come up with a better or the more smoother approximation to this in order to use it together with the differentiable, let's say, neural net generators and whatnot. Although, again, is this the way to go forward? Perhaps, but it's unclear. So that's why what we do is that we build this system so that everyone can explore different bits of the science or the technology, and then making sure that we can actually combine them all together. So we are actually pursuing multiple directions here as well.
I see, very excellent. I just very quick follow-up on this. Like it seems in discrete optimization and multi-objective optimization, genetic or evolutionary algorithm seems to be very effective, and it doesn't require differentiability. Do you have any comments on that, or?
Yeah, absolutely. So if I can say it from two different angles. So in fact, in this crane, the two baselines that I showed you, so NSGA2 and then model-based GA, MBGA, are all genetic algorithms. Now, of course, what is genetic algorithms? Genetic algorithm is, I don't know, I would say it's just a fancier term to say we are doing a very priorless exploration of the space and then choosing the one that is good. Now, priorless exploration has one disadvantage, is that if the space of the exploration is too large, it's unlikely that it's going to work. However, if the exploration space is really low, this lack of the prior allows us to explore that small, narrow vicinity really, really well. So what that means is that if we know what is the right narrow down search space we are working at, genetic algorithm is absolutely excellent. And in fact, some of the success of the genetic algorithms at Prescient Design as a design, although I didn't talk about it, is especially when we are narrowing down or we put a very severe cap on the number of mutations that can be done. And then when we chose the right points for mutation, the genetic algorithm is absolutely fascinating. Now, often the issue is that sometimes we can do that, but many times we don't know what are the actually right amino acids or the residues to replace. So then we have to increase the size of the search space, at which point the genetic algorithm becomes not as effective as many other algorithms.
Great. Do we have any other questions from the room? Or also, if you're attending online, feel free to ask a question in the question and answer panel.
I can ask one if if there's no queue. Thank you so much for the beautiful talk. Really amazing. I have some background in chemical and biomolecular systems, so this is definitely touching my heart. So I appreciate all the details on this multi-objective optimization and machine learning and so on. One particular question I'm very curious about is that when we think about broader AI, I think that the whole system you have is in the lab, in the loop. Basically, we have this machine learning, and then we try to validate it by experiment, which makes perfect sense. But at some point, you did mention that laboratory validation is very expensive, very time consuming. So I'm curious whether you consider anything like digital twin. It's more like simulation model. People talk about quantum chemistry calculation, molecular dynamics. That's more like intermediate state. Of course, that's not going to capture everything you will validate by experiment, but potentially that's going to make everything in silico and hopefully speed up optimization and representation. Was there something ever you try? And if not, or did you just directly do the optimization and the validation, or is there some intermediate steps out there? Professor
Sorry,
Toby.
yes. Okay, there you go. Yes. Yeah, absolutely. That's a great question. Now, as you said, it is indeed very difficult to build a in silico simulation of this kind of biomolecular space or the biomolecules that works at a scale that makes sense for this kind of therapeutic design. It's very difficult to do so. However, we do believe that that is one interesting direction that we must pursue anyway, because it's another thing that the multi-objective optimization tells us, that we need to hedge our bets because we don't know what the right balance is. So in fact, one of the reasons why our team is so large relative to the number of the algorithms or the aspect that I told you about is because we are not just putting all the eggs in this machine learning basket, or we're not putting all the eggs in just sequence only design basket, but we actually do have a lot of people who are working on, for instance, molecular dynamics, as well as the various other more first principle based simulation and simulation based designs as well. So you're absolutely correct. I just didn't talk about it because I'm really not qualified to even talk about those things. However, we do try to ensure that we cover many different aspects, in particular from the perspective of the research. Yes.
Thank you. And if I may, just one quick follow-up question, maybe getting to antibody. So you mentioned sequence optimization many times, and which makes sense, right? I mean, for protein, even for peptide, I'm sure you know peptide, it's a sequence problem, right? And have you ever thought about tries to, I saw you have done a lot of work on, language models, right? I mean, have you ever tried to use some probably it's a very intuitive idea. Why don't you use language model to optimize? I know it's much harder when you get to these 3D conformation, structure interactions, and so on. But do you see the good opportunity to use some sort of, foundation model or even some sort of, like foundation model for these kind of systems in general? Yeah.
Yeah, absolutely. I mean, using language models, many of our property predictors especially are built on top of these pre-trained protein language models, or the more larger, let's say, protein and language models that we build ourselves. And then often doing so helps. Although the effect that we see of using this kind of large-scale pre-trained model has been smaller than what I observed on the language domain. So perhaps we need to do a bit more on that one. However, but that said, at Prescient Design, we are actually training our own large-scale language models from scratch using our own proprietary data that includes both language, various academic publications, internal reports, FDA reports, as well as the various sequence database. And then the reason why we are training them is not because they are large or they're going to give us a better property predictors, but because these language models are amazing integrator of different data. So if I have a heterogeneous sources of information and I want to make connections across all those different sources, these large-scale language models are the ways to go, or at least the only way we know how to go about. And then that's really interesting because if you're looking at the screen now, what we need, how do we backpropagate the predicted clinical outcome all the way to molecular design or the target discovery? We are going to use these language models as the pipe through which we backpropagate this clinical outcome, because these language models can and are being trained on all the data created from all these different stages. So it can learn to make connections and thereby backpropagate these clinical outcomes. So in that sense, the answer is yes, very, very exciting, huge potential there. But what is the right way to go about? We don't know yet. And then there's actually a question on QA, right? Yes.
Yeah, I was just going to read it out for everyone. So in some cases, the property predictor can guide the generative model to generate more reasonable candidates, but in other scenarios, you have very limited samples to train the ML classifier. In these cases, can you use active learning to label some of the generated candidates? Do you think this makes sense as an approach?
Oh, not only makes sense, that's the only way to go about. And whoever asked the question is absolutely correct. We almost never have enough examples. In fact, we almost always start with 0 to 5 examples. And as you can imagine, 5 examples are never going to be enough to train any reasonable predictors because the generalization just won't work. So in this lab-in-the-loop design, this red arrow going from design DB to property predictor is also the arrow that represents training. So whatever comes back from the lab will be used to train these property predictors. So for instance, in the case of the Lambo, let me show you here. In the case of the Lambo, what happened is that by this point, round 12, we were using all the data points that have been accumulated, not throughout the history of the company or the humanity, but also from all the previous rounds of the lab-in-the-loop design as well. So, and then that is how we actually make sure that our systems are going to generalize to the vicinity of what we have seen so far, so that it provides us a good enough signal to ensure that we push forward on all those objectives. So the answer is yes, active learning. Lab-in-the-loop is the active learning platform, and then that's what we're doing already.
Wonderful. Thank you so much. Well, it looks like we're quite a bit over, so how about we thank our speaker one more time. Thanks again for the wonderful talk.
Thank you very much. Thanks for the invitation.
Thank you very much. It's amazing.